So for the next presenter, we will have Rodrigo Gonzalez from the United States of America. He is currently at Harvard Medical School. Um, he is going to take us through the constitutive immunology surveillance of nasal mucosa by neutrophil subsets with distinct origin, phenotype, and function. Welcome, Dr. Rodriguez.
Thank you very much. Thank you. Can you hear me well?
Yes, we can hear you well.
Okay, perfect. So today I'm going to talk to you a little bit about the nasal mucosa, which is an organ that maybe most of us are not familiar with. And I will start by telling you that the nose, as I said, is an understudied organ, but that one that has multiple functions. We're mostly familiar with olfaction and maybe air conditioning. However, I'm very interested in its function as a filter. And this is as a filter of airborne pathogens. These pathogens typically will be trapped in the nasal mucosa and they will be eliminated through mucociliary clearance. However, many of them have evolved to cause local infection. And the problem is that they can further disseminate into the lungs through the airways or even into the brain through the cribriform plate. The question that I'm very interested in answering is, what are the immune cells that guard the nose and how they interact with pathogens? And to answer this question, I designed an intraviral model to track the dynamics of cells in the nasal mucosa. And the most interesting finding can be summarized in this video that I'm going to show you right now. And what you see here is the nasal mucosa with neutrophils in green and blood vasculature in white. What I want you to notice is the large number of neutrophils that are in the extravascular space of the tissue. And this is interesting because this is an untreated animal. And what do we know about neutrophils in general? Well, we believe that neutrophils are at the forefront of infection and trauma. Importantly, that they migrate from blood into tissues only when homeostasis is disrupted. Maybe for this reason, they're rarely studied in the steady state. We also know that they engulf pathogens and they possess high antimicrobial abilities. And we also think that they are terminally differentiated and they are short-lived. So in the context of what I just told you, it's interesting to think that we see these extravascular neutrophils in the tissue that I imaged. However, you can argue that because this is surgery to access the tissue for a video, I could have damaged the tissue, and this is what brought the neutrophils. However, we can still see them in fixed tissues of mice that have been undisturbed at all, and here neutrophils are in red and blood vasculature is in green. We see them in large numbers that actually account for about 25% of all extravascular immune cells in the tissue. Interestingly, not only in conventional mice, but also in germ-free mice. Overall, what this is telling us is that neutrophils are constitutively present in the nasal mucosa, not because of trauma and not because of the flora that is present in this tissue. So the question that we asked next was, what are the characteristics of these neutrophils? And we can easily identify neutrophils in blood as a single population using the markers Ly6G acetyl11B through flow cytometry. However, when we look at the nose, we can see that neutrophils can be identified as two populations based on the same markers, one that is high and one that is intermediate for the two markers. If we zoom in into these populations, we see that the high population can be further divided into two populations based on the markers CD11C and cyclic F, which are not typically found in neutrophils. Thus, we have three populations in the nose, three populations of neutrophils. First, one that we term N1, which is intermediate for Ly6G and CD11B. Second, a population that we term N2, which is high for the two markers. And third, a population that we term N3, which is also high for the same two markers, but now this one is double positive for CD11C and cyclic F. Especially considering that these two markers, CD11C and cyclic F, are not common in neutrophils, we ask the question, are these truly neutrophils and how similar they are to other conventional neutrophils? And when we look at the nuclei of the three populations, they are comparable to the nucleus with a polymorphonuclear profile of conventional neutrophils such as those from blood. Moreover, if we do bulk RNA sequencing, we see that the three populations are different from bone marrow and blood neutrophils, but they're also different from one another. So the next question that we ask is, do blood neutrophils enter the nose through differentiate them into the three subsets? And to answer this question, we use parabiosis. Parabiosis is a strategy normally used to track entrance from blood where the circulatory system of two mice with different congenic markers is joined. These congenic markers, I will refer to them here simply as blue and green. And if we look at blood circulation of the blue mouse after 1 month of being conjoined to the green mouse, we see that there is comparable percentages of blue versus green neutrophils, which makes sense because the circulatory system is conjoined. If now we look at the nose of the blue mouse and we track the N2 and N3 populations, we see that the percentages of neutrophils is similar to what we found in blood. No surprise, this is consistent with the blood origin. However, if we look at the N1 population, now in the blue mouse again, we see that there's a prominent population of blue neutrophils and not green neutrophils, which suggests a local origin, which is very unexpected because as I described before, we believe that neutrophils come from blood into tissues. So the question that we wanted to answer then is, where do N1 neutrophils come from? Tracing the tissue, we see that the entire nasal cavity is full of compartments that seem to have bone marrow. Indeed, with the lice MGFP mouse, where neutrophils are bright green, we can identify these pockets of bone marrow all over the place. So then we said, well, maybe what's happening is that we are seeing the N1 population, not necessarily as a nose population, but simply as bone marrow neutrophils. Indeed, when the frequency of the N1 population is markedly reduced in preparations that do not include bone. So this is mucosa and bone. We see the N1 population. This is mucosa only. The N1 population disappears. So is it as simple as contamination from bone? Well, when we looked directly through imaging, we can identify the bone marrow pockets where the N1 neutrophils reside. We can identify mucosa and the bone that separates these two compartments. However, if we look carefully, we see channels that connect these two compartments. Not common, rarely, but we do see it. There's neutrophils that actually can be present inside of these two channels, opening the possibility of flow of neutrophils from bone marrow into the mucosa. Under specific conditions, one in particular, after treatment of methimazole, which is a drug that is used to deplete olfactory neurons, we see a great increase in the flux of neutrophils from one compartment into the other. Overall, then, this indicates that N1 neutrophils survive in the bone marrow, but they are connected through direct channels into the nasal mucosa, and that under specific conditions, there could be an influx of these neutrophils from one compartment into the other. But what about N2s and N3 neutrophils, which we already know that enter for blood? Do they do so as a separate population? We did single-cell RNA sequencing of these extravascular neutrophils, and we label them with barcoded antibodies with the same markers that I used to identify the populations. The purpose of this is that aside of identifying different clusters, in this case, we resolved 7 different clusters, these clusters can be linked to neutrophil subsets based on protein expression in such a way that we have clusters for the N1, N2, and N3 populations. Pseudotime analysis delineates a linear pathway, a differentiation pathway from N1 to N2 to N3. It makes sense for N1 as they are in bone marrow, so they're the youngest neutrophils. However, we don't know in the case of N2s actually differentiating into N3s. To test this, we injected fluorescently labeled neutrophils from bone marrow or from blood into blood in an animal that is not labeled. And we can find these neutrophils here labeled in red as early as 5 hours after injection with a profile that is double negative for cyclic F or CD11C, which is consistent with the N2 profile. And it's only after 6 days that we see an increase in these two markers to develop a fully N3 profile consistent with the N3 phenotype. This is not present in bone marrow or in blood, and this overall suggests that indeed neutrophils enter from blood, they differentiate into N2 neutrophils, and only after 6 days, they differentiate into N3 neutrophils. The most important question is, are there any functional differences that correspond to these populations? And we know that these neutrophils can, as any neutrophil, eat. We saw them eating pathogens, in this case, Candida albicans, and we wanted to know if one population is better than the other at doing this. To our surprise, we found that it's only the N2 population that actually engulfs pathogens, not only Chlamydia myoidarum, but also Streptococcus pneumoniae and also Candida albicans. So it's a neutrophil-dependent phenomenon and not a pathogen-dependent phenomenon. But what about the N3 neutrophils, which didn't seem to catch pathogens? Well, we still don't know, but we have some interesting data. First, we know that N3 neutrophils seem to downregulate all genes that are ascribed to neutrophils. So it's as if the identity of a neutrophil pretty much changed and disappeared. They also upregulate genes that are ascribed to dendritic cells and macrophages. And if you look at the morphology, they acquire N3, a dendritic morphology, while N2 neutrophils keep a more or less amoeboid morphology. This indicates that not only N2 differentiate into N3, but the cellular identity changes into something that we could call maybe an antigen-presenting cell. So the next logical question to ask is, can N3 neutrophils actually act as antigen-presenting cells? And to answer this question, we sorted neutrophils, we pulsed them with OVA for 3 hours, washed them, labeled OT1 and OT2 cells with a proliferation dye, and cultured them with neutrophils for 3 days. And to our surprise, it was only the CD8 T cells that proliferated only in the presence of the N3 population that had seen OVA. This is concomitant with upregulation of CD69, CD44, CD25, and glycine B, and downregulation of CD62L. Overall, what this means is that N3s are able to present antigens specifically to CD8 T cells. This is a phenomenon known as cross-presentation that is typically described or only expected to be present in a subset of the dendritic cells called CDC1s and not in neutrophils, which we think is an exciting finding. Overall, we have then 3 different populations. N1 that reside in bone marrow, they had direct access to the nasal mucosa. Meanwhile, blood neutrophils enter to the nasal mucosa to differentiate into N2 neutrophils, which during infection can exit the mucosa, go into luminal space, and kill pathogens. In the steady state, however, these neutrophils will differentiate into N3 neutrophils in time in more or less 5 to 6 days, and they will acquire abilities to present antigen to T cells. I didn't show you this data, but they will not recognize pathogens. They are not attracted to pathogens, and they will collect material from surrounding cells. This is all in contradiction of traditional views of neutrophils, which describe them as recruited to inflame tissues upon infection and trauma, short-lived, and terminally differentiated. And with this, I want to thank the rest of the people in my lab, and I'll be more than happy to take any questions.